Analysis of lipids by Sudan 3 test. Definition. The Sudan 3 test is a dye binding assay used to identify the presence of lipids, fats, and oils. Sudan 3 is a fat-soluble, lipophilic dye that stains lipids a distinct reddish-orange color because it dissolves more easily in nonpolar lipids than in the polar solvent used for the dye. 1. Experiment. To detect the presence of lipids in a food or chemical sample using the Sudan 3 staining method. 2. Apparatus. Test tubes, test tube rack, and droppers. 3. Chemicals. Lipid sample e.g., vegetable oil, distilled water, for control, and Sudan 3 dye solution. 4. Procedure. Take 2 milliliters of distilled water in a test tube and add 2 milliliters of the lipid sample. Note that the oil and water form 2 separate layers, oil on top. Add 3 to 5 drops of Sudan 3 dye to the test tube. Shake the test tube gently to mix the dye with the contents. Allow the tube to stand undisturbed for a few minutes in a rack. Observe which layer absorbs the dye and changes color. 5. Precaution. Sudan 3 can stain clothing and skin. Handle with care. Ensure the dye is well prepared in an alcohol, water base to ensure proper solubility. 6. Observation. The dye concentrates in the top layer, the lipid layer, staining it a bright reddish orange. The bottom water layer remains relatively clear or only slightly tinted. 7. Result. The selective staining of the oil layer confirms the presence of lipids. 8. Formula. Lipid plus Sudan 3 dye converted to red stained lipid layer. Since Sudan 3 is nonpolar, it migrates from the solvent into the fatty acid chains of the lipid. Asterisk. 9. Formula use and solve mathematical numbers. If you are testing a asterisk 10 milliliters sample of milk that is found to contain asterisk 4% fat, calculate the volume V of actual fat present. V equals total volume times percentage 100. V equals 10 times 4. Answer. The milk sample contains asterisk 0.4 milliliters of pure fat. 10. Diagram. Draw a test tube showing 2 layers, a clear bottom layer and a colored top layer. Color the top layer reddish orange and label it as the lipid layer. Label the clear bottom layer as the water layer. Indicate that the dye is only soluble in the fat.